Hi, everybody. My name is Rich Condit, and I am a virologist. That is, a person who studies viruses, which I've been doing for about 50 years. During the pandemic, I've had an opportunity to engage in numerous educational outreach activities, some of which were targeted towards the general public, and I've become aware of a need for a presentation that could explain the basics of viruses, of vaccines, and of variants, with particular reference to SARS-CoV-2 and COVID, to the general public. And so this is my attempt to do so. Welcome. So here's an outline of our journey. First, I want to describe what a virus actually is. Since viruses hack the machinery of cells, I have to start by telling you how cells work. And then I can tell you how viruses work. With that information in hand, I can give a little more specific information on the virus that causes COVID-19, that is SARS-CoV-2. With that in hand, I can then proceed to describe the various technology platforms that are used as virus vaccines and how they work. And lastly, I'll discuss a little bit about the virus variants that people have heard of. So first, what is a virus? So, how do cells work? For a person of my training, this involves describing what we call the central dogma, which is the flow of information from DNA, the informational molecule in cells, to protein, which is the workforce in cells, all of which I will describe. Shown in this diagram is a rectangle that's meant to represent a cell. I believe that all people, or most people, are familiar with the concept that the cell is the fundamental unit of life. Organisms are composed of many cells. Humans contain, I think, on the order of 1 trillion cells. And this one cell, the interior, is colored yellow, and that's a watery or aqueous environment. And is surrounded by this gray boundary, which is made of a chemical called lipid, which has a sort of a greasy chemical nature so that it doesn't mix with the watery interior of the cell, and therefore provides an ideal boundary to separate one cell from another. Here's a DNA molecule contained in the cell. I believe most people are familiar with DNA as the genetic information, usually represented more accurately chemically as a double helix. But for my purposes, it's convenient to represent it as this string of boxes. In this string of boxes, each box is meant to represent a gene. Each gene, for our purposes, encodes a protein. The entire complement of genes that is necessary to provide the instructions for the development of an organism from a fertilized egg up to an adult, and the differentiation and function of all of the different tissues in that organism, heart, kidney, lungs, is encoded in the DNA molecule. So that's quite a bit of information. In humans, this is encoded in 23 pairs or 46 chromosomes, comprising about 30,000 genes. And importantly, in all organisms, in every cell in the organism, from the fertilized egg up through development and in the adult, every cell contains the entire genome, the full complement of DNA necessary to do the whole job, from fertilization up to functioning adult. And in fact, when cells duplicate, the DNA molecule is copied into identical molecules, the cells divide, and each daughter cell inherits the entire genome. But at any given time during development, or in any given tissue in an adult organism, one only needs a fraction of that full complement of genes. And this is accomplished through a process called transcription. During transcription, individual genes are copied into a molecule of very similar chemistry called RNA. So the chemistry is so similar that the precise code in DNA is copied and replicated as this RNA molecule. And note in this description that even though I have 10 genes in this genome, in this particular cell, I've copied only 3 of those into RNA for the immediate purposes in that cell. Note also that I can make different amounts of each different gene. I've made 3 copies of this red gene, 2 copies of this blue gene, 4 copies of the green gene. I want to point out at this point that there are actually several different functional classes of RNA in a cell. This is one major class. It carries the information from DNA in the process of making protein, and so is called messenger RNA or mRNA. I'm not going to talk about the other classes, and I will use the words RNA and messenger RNA almost interchangeably. I also want to point out that RNA is a temporary molecule. I may want to turn genes on and off, and so it's just as important to get rid of RNA as it is to make it. So whereas DNA is a permanent record, RNA or messenger RNA is a temporary copy or message from the DNA. This information then gets translated into protein. Now, proteins are, as I said, the workforce of the cell. Proteins carry out all the chemical reactions. They provide the structure, or most of the structure. They provide a lot of the mechanics that make a cell function. They are a very different chemistry than RNA or DNA, which, as I said, are very similar. Proteins are the fundamental primary chemistry from one protein to another are quite similar, but in a full-length protein that's folded up to make the finished structure, one protein can look quite different from another, and I'll give some examples of this as we go along. So I've represented these as different from RNA or DNA and different from each other. So I got circles for the red gene, ovals for the blue gene, triangles for the green gene. So that's how cells work. So here we have an idealized virus. The virus, any virus, consists minimally of a shell inside of which is contained the virus genome, which can be RNA or DNA. And we'll go through this in a little more detail. The shell can be called a capsid or an envelope. If it's made entirely of protein, it's a capsid. If it contains a lipid bilayer, it's an envelope. I've represented here an envelope where the gray would be a lipid bilayer similar to the boundary, the cell membrane, the boundary around cells. And I've embedded in that a virus-coded protein. That's the virus attachment protein. Inside the virion or virus particle, inside the shell or envelope, is the viral genome. The genome can be RNA or DNA, and in fact, viruses are the only organisms that can use RNA as the informational molecule. All other organisms use exclusively DNA, and it depends on which virus family you're talking about, whether it uses RNA or DNA. In my idealized virus, I've shown it to have just 3 genes. All viruses have to encode a protein that is required for replication of the virus genome. So I've labeled that R, and its job is to bring about copying this into multiple copies of the genome during growth of the virus. All viruses also encode some sort of a structural protein that makes up the capsid or is embedded in the envelope. In the case of SARS-CoV-2, this is the spike protein. And if there's at least one of these in any given virus, there can be multiple, and at least one of them will serve as the virus attachment protein as well. As viruses replicate, they have to contend with what can be a hostile environment. Cells have defense mechanisms against viruses, Organisms have defense mechanisms, that is the immune system against viruses, and the virus may want to tweak the intracellular environment in the cell or the environment in the organism to overcome some of these defenses and bring about a better environment for it to replicate. And in the process of tweaking the cells or the organism, one side effect is that it can make you sick. So I've labeled this as a virulence gene with a V. So there's our idealized virus. So now let's look how a virus infects a cell. Here's our virus. Here's our cell going about its business. The virus attaches to the cell, and this can be a very specific interaction between the virus attachment protein on the surface of the cell and a protein that's embedded in the membrane of the cell. In the case of SARS-CoV-2, you may have heard that the receptor on the cell surface is a protein called ACE2. Having bound to the cell, the virus can then be taken up inside the cell. It's then uncoated, and the virus genome is released into the cell. And since this particular virus that I'm showing here uses RNA as its genome, that RNA molecule can be inserted directly into the messenger RNA pool of the cell and translated into viral proteins. So it uses the cell machinery to translate this replication gene into replication proteins, the virulence gene into virulence proteins, the structural gene into structural proteins. Note that DNA viruses follow a very similar process, but since their genome is composed of DNA, they insert themselves into the normal processes of the cells upstream from here and must be transcribed into messenger RNA in order to make viral proteins. Following this, then, the replication protein does its job and serves to make multiple copies of the virus genome. So we have several copies here. Those can serve as templates to make much more of the structural protein and some of the virulence protein, which may tinker with the environment of the cell to assist in this whole process. All that material then can assemble into new virus particles, which then somehow escapes the cell, quite often in the process destroying the cell. And so that's how a virus works. I'm showing this slide just to give an idea of the diversity of viruses in nature. These are just a few of the virus taxa or virus families that infect vertebrate organisms. DNA viruses on the top, RNA viruses on the bottom. These are shown in relative size. So you can see there's quite a diversity in size and quite a diversity in form for different viruses. Viruses infect virtually all forms of life: vertebrates, invertebrates, fungi, plants, bacteria. There are hundreds, if not thousands, of different virus families. They're extraordinarily abundant in the biosphere. Some of them cause us harm. Some of them, many of them, are completely innocuous and just live with us. Others may even create some benefit in us or in the biosphere. So these are fundamental components of life on Earth. So now that we know a little bit about viruses, we can move on more specifically to SARS-CoV-2. I want to get the vocabulary straight to start with. The virus that causes COVID-19 is called SARS-CoV-2 for severe acute respiratory syndrome, coronavirus 2, and the 2 distinguishes it from SARS-CoV-1 or SARS-CoV, which is a closely related but not identical virus that broke out in, as many will recall, 2003, caused about 8,000 documented infections and killed about 800 people. You might ask, logically why SARS-CoV-1 was limited while SARS-CoV-2 caused a pandemic. And one of the major reasons, in fact, is because SARS-CoV-1, by and large, did not cause asymptomatic infections. So you could identify individuals who could spread the virus and isolate them while they were sick, whereas a large fraction of the SARS-CoV-2 infections spread through infections that cause no symptoms or spread before symptoms emerge, which makes it much more difficult to contain. The disease caused by SARS-CoV-2 is called COVID-19, for coronavirus disease, that arose in the year 2019. So here we have a diagram of SARS-CoV-2 on the left and an electron micrograph of the real thing on the right. Go through the diagram a little bit. This is an enveloped virus, and you can see a sort of a blue-gray material in the envelope here, which represents the lipid membrane. There are actually 3 proteins embedded in the membrane. This brown one here called the membrane protein, this orange one here called the small envelope protein, and most importantly, this large yellow protein sticking out of the virus called the spike, which for those of you who have been following the pandemic may be familiar with. The spike is the viral attachment protein that it uses to attach to cells, and it's also, as we will see, the primary component of vaccines against the virus. Because as you might imagine, if you can somehow compromise the function of that spike protein, you might compromise the function of the virus. Inside this particle is the RNA genome that's encased in yet another protein called nucleoprotein. Over on the right is the electron micrograph. As you can see, it's round or sort of blob-shaped, and sticking out of it, you can see these individual spikes. Those are the individual spike protein. And the first people who described this saw a resemblance to a crown or to the sun's corona, hence the name coronavirus. Here is the SARS-CoV-2 genome in detail. The genome is on the top here, and there's some detail about how the genes are expressed shown in the lower part of the slide. Of course, I don't expect you to assimilate all this, but I want to use it to make the point that my model virus is grossly oversimplified. This RNA genome shown on the top is 30,000 units or nucleotides in length. On average, you need about 1,000 units to encode a gene or to comprise a gene or encode a protein. So that would be about 30 genes, and in fact, SARS-CoV-2 comprises about 30 genes. Genes are represented here as boxes in the genome and subdivided into more boxes in this portion down here. The left-hand 2/3 of SARS-CoV-2 encodes primarily replication proteins. These are actually translated as one big long protein that then is cleaved into individual proteins shown in these individual boxes. The right-hand 1/3 encodes primarily structural proteins, and here you can see the gene for the spike protein uses the same color coding as I showed in the previous slide, the envelope protein, the membrane protein, and the nucleocapsid protein, and these are individual messenger RNAs that are made from this region that encode each of the individual proteins. There are numerous other proteins here, many of them shown in pink, that for our purposes, some of these could be virulence factors. Many of these are proteins that alter the environment of the cell or compromise the immune response of the host to the advantage of the virus, and in the process could make you sick. So now we know a little bit about SARS-CoV-2, and we can move on to vaccines. With this slide, I'm going to describe the 5 basic platforms that comprise viral vaccines currently in use, in particular in the context of the COVID pandemic. This gets a little detailed and it's going to take a little while, but hopefully it will be understandable, and it is the sort of crux of this whole presentation, so bear with me. You will be rewarded. So the first of these platforms is what we call an activated virus. And this involves growing up a large amount of the virus that actually causes the disease and treating it with a chemical that reacts with the RNA or DNA that serves as genome and has the effect of inactivating those genes, which I've represented here as X's over the replication, the virulence, and the structural gene. So now we have a particle that still looks like the virus, but can no longer replicate. And if I inject this into a recipient, hopefully they'll mount an immune response to the particle and be immune to future encounters with the virus. A more subtle technology is what we call an attenuated virus. And in this case, all I've done is to inactivate the virulence gene, leaving the replication and the structural genes intact. There are a number of different ways of doing this. The traditional way is to take this virus and grow it repeatedly through repeated growth cycles, either in cell culture or in fertilized chicken eggs on chick embryos. This can be for hundreds of passages. And under these conditions, the virus doesn't have to compete with many of the normal antiviral activities of the immune system or in the cells it ordinarily encounters, and it gets kind of lazy and loses some of these virulence factors that are designed to cope with those challenges. So now we have a virus that no longer has these virulence genes. Nowadays, we can actually identify candidate virulence genes just by looking at the genetic code for the virus and use genetic engineering techniques to specifically inactivate those and make attenuated viruses. But it's still pretty tricky. At any rate, if successful, what we have now is a virus that after injection into a recipient will actually make more viruses using the intact replication and structural genes, but it won't make you sick. And because we get an amplification of the viruses and make more gene products, this may have some advantages over the inactivated virus. A third type is a protein vaccine. And in this case, we simply identify what we believe will be an appropriate immunogen. I've represented that here as this structural protein that we think if we could make an antibody to that, we could confer immunity. And using genetic engineering techniques, simply make a large amount of just that protein and inject it into a recipient. Hopefully they'll mount an immune response to that and be immune to infection. Now I'll describe two more modern technologies. The first of these is the viral vector vaccine. In this case, we've taken a completely different virus, represented here by this hexagon with these black triangular structural virus attachment proteins on the outside. That virus has its own replication gene, its own virulence genes, its own structural genes, and I've done two things. First, inactivate the virulence gene so that a recipient infected with this viral vector can't be made sick by this virus. In fact, a lot of these viral vectors don't even replicate. So under the circumstances of injection with these, they don't even use the replication or structural genes. The second thing I've done with this is to, using genetic engineering techniques, splice into its genome the gene for the structural, that encodes the structural protein for the virus that I want to immunize against. So that this is now a very effective gene delivery vehicle. If I inject this into a recipient, they will take this up and hopefully make the protein encoded by the structural gene mount an immune response to that and confer immunity to infection by the virus from which this gene arose. The last vaccine platform is simply a messenger RNA molecule. Or in fact, you can use a DNA molecule, but most of the vaccines in use use messenger RNA, which is just the coding sequence for the structural protein that I think will confer immunity, delivered in a nanoparticle composed of primarily lipid, the same sort of material that exists in the envelope of the virus or on the outside of cells. This protects it from degradation during delivery and helps in its uptake. There's an important distinction to be made amongst these five. These two, protein and inactivated virus, involve preformed protein. That is protein that's formed before injection. So you're injecting protein. No protein is made during the immunization process. In these three cases, the protein of interest is actually produced after infection using the cellular machinery, and I will detail this for the viral vector and the messenger RNA vaccine in the next slides. There are some differences in how one responds to these two technologies, but for our purposes, they are similar. But I just wanted to make that distinction so people are aware of it. In any case, the end result of administering any of these is to make protein for the immunogen of interest. In this case, or in the case of SARS-CoV-2, it would be the spike protein. Now, your body, as many people know, can respond to this as a foreign object by making antibody to it. Antibodies are also proteins made by your cells, in fact, made by specialized cells in your body and designed to attach to the foreign protein very tightly and very specifically, discriminating it from all other proteins. Now, this actually takes a while because your body has to sort through a number of different candidate cells that might be able to produce the appropriate antibody, and then ramp up production of that antibody, amplify the number of cells to make that antibody, mature the affinity of antibody for the protein. But once you're all set, you now have a bunch of antibodies so that if you encounter this protein in the future, the antibody can react to it. So let's have a closer look at the mRNA vaccines and how they work. And just as a side note, the two mRNA vaccines that are in use for COVID currently, there are others in the pipeline, are those made by Pfizer and Moderna. So what happens is that one injects the messenger RNA vaccine, it's taken up by cells and plugged right into the central dogma as messenger RNA, and the cellular machinery then translates that messenger RNA into virus protein, into this spike protein. That protein can be presented on this, depending on the circumstances, the surface of the cell, or actually extruded from the cell. And in either case, one can make antibody to that spike protein, and that's your immune response. So let's have a closer look at how the viral vector vaccine works. And in the context, the current context of the pandemic, the viral vector vaccines that people may be familiar with are those made by Johnson & Johnson, which is the same as Janssen, and another made by AstraZeneca. So in this case, one's injected with the viral vector vaccine, which then infects cells, and the genome is then released from the virus particle, and I've shown here a DNA-containing viral vector. So the genome is inserted into the central dogma at the level of DNA. As I said before, the replication and structural genes of the vector may or may not be actually expressed. This may or may not replicate to some extent. The current vaccines have no replication of the vector genome. Transcription occurs, and in this case, just the messenger RNA for the structural protein, or in the case of coronavirus, the spike protein, is made. This is translated into protein, which then can be expressed on the surface of the cell or secreted from the cell, and one elaborates antibody to that protein. And I want to emphasize that in the case of all of these vaccines that involve uptake of a template, be it messenger RNA or an attenuated virus or a vectored vaccine, and synthesis of the protein in the cells, there's only a relatively small number of cells that are actually involved in synthesis of the protein. And some of those cells even are some of the most important of those cells are cells that are actually specifically designed to participate in the immune response. And all of these cells ultimately are turned over and replaced. And so there is no permanent change done to the organism aside from the fact that you've mounted an immune response. So now we have a lot of antibody that's very specific for the SARS-CoV-2 virus or whatever virus we've used as an immunogen, so that when we encounter that again, hopefully the antibody will react strongly with the surface protein on the virus, compromise its function so that the virus can not infect cells. Now, as I said, this initial immune response takes a little while to sort out the cells, amplify them, mature the antibodies. So if we look at the concentration of antibody over time after the initial exposure, it takes a little while and we get an immune response. And many of these vaccines require a boost. And what happens there is there's a secondary exposure during which you get a much faster immune response, many more antibodies, and higher affinity antibodies. So that's the effect of a boost and why it is used under many circumstances. Now, I've significantly oversimplified this. There are many complexities. Importantly, there are many different antibodies that can be elaborated against any given protein. So while I've shown just one here interacting with the tip of the spike, in reality, there may be dozens of different antibodies with different specificities that can react with different parts of the spike protein. And that becomes important, in particular when we start to consider variants. Also, during a natural infection, there are many viral proteins made, as you've already seen in the discussion of SARS-CoV-2. So, in fact, during a natural infection, there may be other immunogens that that you make an immune response to that help in conferring immunity. And lastly, I've talked only about the antibody response. There's another whole arm of the immune response involving cells called T cells that assist in conferring immunity. So, with that information about vaccines, I now want to say a few words about variants. And variants involves mutation of the genetic material of the virus. That is a change in the coding sequence of the genes of the virus. Mutation can happen in any gene, the R gene, the V gene, the S gene, and the possible consequences are infinite, from no consequence at all to actually making it so the virus doesn't work at all. But you could also, on rare occasions, perhaps introduce mutations that potentially could increase the ability of the virus to spread, potentially could increase its virulence, potentially allow the virus to escape existing immunity. Recall, however, that immunity is very complex, and so it's not a simple matter to escape immunity, and one mutation may not do the trick. I've represented mutation here as just a black dot, for example, on this structural gene that when decoded could be represented as a black dot on the tip of this. As an example of how this might function in, for example, the escape of an immune response, it could be that this causes a structural change in this protein so that the antibodies that you made to the original virus can no longer fit properly onto the end of this altered protein. So they might not work as well anymore. But recall once again that the immunity is complex. There may be many other antibodies that react with this protein, and there's also the T cell response. So evading immunity is not a quick or simple process. It may require quite a few mutations. So there you have it. Viruses, vaccines, variants. I want to point out a few additional resources. The go-to resource is the Centers for Disease Control, which has all the latest updated information on the disease and on the virus and vaccines. If you're interested in the particular vaccines that are being developed, the New York Times Coronavirus Vaccine Tracker I have found to be absolutely excellent. And you can find what the latest vaccine candidates are, how they were manufactured, how they work, what they do, which of the virus platforms or which of the vaccine platforms they use. And it's all quite understandable and very well done. It's a great resource. Children's Hospital of Philadelphia Vaccine Education Center has a lot of information on vaccines, availability, how they're administered, and in particular is a great resource for understanding vaccine safety. If you have any questions and would like to ask an expert, the American Society for Virology COVID-19 Vaccine Education Town Halls are available to you at this link, asv.org/education. Here you can meet for 45 minutes in an online format with 2 out of a pool of 50 experts, individuals such as myself, who will receive your questions about vaccines, how they work, or vaccine safety, and do their best to answer them. They're free. There are numerous town halls available each week. You can come back as many times as you want, and I encourage you to take advantage of that resource. Lastly, if this sort of thing interests you and you want to get further into the weeds, I can recommend to you a podcast on which I am a co-host, run by Vincent Racaniello, called This Week in Virology at microbe.tv/twiv. Hope to see you there. So I hope you found this useful, and have a good day.